Hi everyone, welcome to the Vertebrate Division of the Biodiversity Research Collections here at the University of Connecticut, the BRC as we call it for short. I'm Dr. Erin Kuprowitz. I'm the Vertebrate Collections Manager, so I'm in charge of taking care of all of our specimens that have bones basically. So mammals, birds, herpetofauna, and fishes too. Today we're just going to take a little walkthrough basically. I'm going to show you different ways of handling different types of specimens that we have here and kind of tell you a little bit about each of the things that we have. First off, I just want to say you'll probably notice my mask. We're in the midst of the COVID-19 pandemic. The museum isn't quite open as it usually is these days. We have to take really strict health regulations into account, but the managers do come in from time to time to take care of the things that we have here. We're actually at this point, I think, 140 days since we were shut down. So we're still operating in a diminished capacity, but taking care of the things that we have here. I'm going to talk a little bit about handling specimens first off. So you'll tell also that I have gloves on. Whenever you're handling any type of vertebrate specimen, you always have to wear gloves, no matter what type of preparation it is. This is to protect the specimens from oils and things that might be on your hands that could degrade them over time. And it's also to protect you. So a lot of the specimens that we have in the BRC, in the vertebrate collection, were from kind of prior to the 1970s, and they were prepared with potentially really toxic chemicals like arsenic, mercury, things like that. So it's also for your own protection. So even handling things like bones, we really want to take care of them, and you always wear gloves on your hands. You're only going to be picking up, if you have to actually pick up a specimen to study it, to measure it, you pick up one at a time. You're not going to hold multiple skulls in different hands. You always want to support the specimen with one or two hands, however you need to, and don't pick things up by their most fragile parts, like bones in particular. You're not going to pick up a bird by its leg. And we definitely don't pick things up by their tags and things like that. We'll kind of get to that later with the round skins. You just want to be very conscientious. You're not dropping things on the floor. You're not breaking pieces off either when you're handling them. Again, if you're dealing with large specimens, so like big skulls or big round skins, you want to use both hands, or maybe even a friend can help carry things around. And so now we're going to move in and talk about different types of preparations that we have here in the vertebrate collections and kind of why we study different things in different ways. So here you can see a few examples of bones or skeletal preparations that we have. This is one way of preserving, for instance, mammals. We have here birds. That's really common in our collection for these different types of animals. And we have to clean all of the muscle, the tissue, the fat especially, off of skeletons and bones when we put them into the collection. This is so that we don't attract pests, other kind of, especially insects that could attack hides and even other collections, so the plants, the insects as well. To clean all of the goo, as I call it, off of skulls or skeletons, we usually do this with dissecting tools. So we'll just take kind of probes and scissors and forceps and pull all of the meat off as much as we can, basically. We're not super great at that. You could do it mechanically. You can also take very small bones, so things like hummingbirds are just too delicate to use forceps to kind of pick all the meat off. You can use maceration, which is where you put a skeleton in water and wait for the meat to just dissolve, basically, and rot off, and then you can preserve the skeleton that way. Another thing that you can do for skeletal preparations is use insects. So there's a type of beetle in the family Dermestidae, so we call them dermestids, and they're really good at cleaning bones, cleaning skulls, small bones, and they're just very good at their jobs. However, dermestids are also pests of collections. So some museums do keep active colonies of dermestids, and they put skeletal material in to get really cleaned thoroughly by these beetles. However, we don't keep a colony here because they're difficult to contain. If they get out, they could just go crazy in a collection and destroy a lot of your specimens. So we have friends down at Yale University and the Peabody Museum, and they have an active colony that we use if we want to clean our skeletal material. So kind of going into a few examples of bones that we have to show off today. This is one of our kind of poster children here, the Chacoan peccary. So its genus is Catagonus. It was once thought to be extinct, and I'll say that it was thought by scientists to be extinct. Local people in the Chaco area of South America knew this thing existed for many, many years, but in the 1970s, Dr. Ralph Wetzel kind of rediscovered it. So he took a lot of specimens from Paraguay, from Argentina, and the Chaco area, and he brought them back here. And this was one of the foundations of our museum here. So this is a pretty famous example of the mammals that we have. And again, we have a lot of specimens from local areas, so Connecticut, the Northeast, New England, but we also have surprisingly a lot of South American animals that Ralph Wetzel brought back. He was a big player in the BRC in our early days and established a lot of our really nice specimens in kind of the 1970s and 80s. This peccary, so it's a pig-like animal. You can kind of see in the pictures here. So this is the Catagonus. These are the other two species that live today. The Catagonus is the largest of the three, and these guys like to eat vegetation. This one, the Catagonus especially, likes to eat cacti. It's from kind of arid regions, desert regions in the Chaco. And it really loves to eat cactus pads and fruits from the Opuntia genus. So it's still around. It's not as common. It's restricted to a kind of smaller range than the other two species, but we have a lot of examples here. We probably have the best collection of the Chacoan peccary of any museum in the world. And so moving on, we have another skeletal preparation here. We have a skull, and the bones are actually in this little box, so we're not going to open that right now. This is an agouti, so Dasyprocta azere is the species. This is what it looks like in life, so kind of a nice potato-shaped animal. They have these really great incisors that they use to eat fruits and seeds, really hard materials. Agoutis are rodents, so they're only found in Central and South America, mostly in tropical rainforests. And they are really good seed dispersers. So I like to kind of describe them as guinea pigs on stilts. You can't tell too well from this picture, but they have really long legs, but they have really round guinea pig-like bodies. And they also are described as forest gardeners or jungle gardeners because they will take fruits and seeds and they bury them in the soil so that they can eat them later, kind of like what eastern gray squirrels do around here in Connecticut. Sometimes they forget where they've put things, and that seed might germinate and become a new tree. So they're kind of reforesting certain areas of the tropics. A really cool little animal. So moving on still in the skeletal specimens, we have a bird skeleton here. So this little box kind of looks like a bit of a mess. We have all sorts of bones, and you can see that each of the bones in this box is labeled with a catalog number. So someone went through at some point and used a archival ink pen to record the catalog number from that individual on every single bone. So we do this to keep organized and to know which bones came from which individual, basically, when you're doing things like measurements. So these bones came from the tufted puffin. That's this one right here. It's the largest extant or living species of puffin that's around. And this material was actually collected, found dead on the beach in Alaska, and it was salvaged. So when we're talking about vertebrates and we're talking about specimens that we bring into our museum, we only deal with salvaged specimens. So we have a lot of scientific research permits that we keep. Again, working with vertebrates is a little more complicated than maybe plants or insects sometimes. And we basically are only allowed to collect things that we find already dead in good condition. That's the only way that birds and mammals are introduced into the collection these days. We don't have people going out and shooting things or killing things or taking specimens. So we only salvage material and use that in our collections these days. So that kind of does it for the skeletal preparations. So that's one way that we preserve vertebrate specimens here in the BRC. Mostly mammals and birds. We do have some amphibian and reptile and fish skeletons, but that's a much less common way of preparing these things. We'll talk about that a little bit later, but we usually preserve those in ethanol. So moving away from skeletons and bones, we also use round skins, what we call round skins, and I'll talk a little bit about that when we're preserving, again, mostly birds and mammals. That's the most common type of preparation that we have for those groups in our dry collection in this room. So coming back over here, we have a couple of examples of the same species that we saw in the box of bones that I just showed you. These are two examples of round skins of those tufted puffins, again. And so you can see they've basically had all of their insides taken out, all of that goo that I mentioned earlier. So we try to remove all of the fat, all of the oil glands if they have any. But some bones are kept inside, and that kind of varies from group to group. So with birds, we keep their skulls intact, feet bones, and some other bones, depending on the person who's doing the preparation or the intactness of the specimen that we've salvaged. With birds, you always keep the skull because you have a bill attached to it. And you want to see that bill for certain diagnostic characters and for certain traits that you look at. And so once you've taken out all the insides, you kind of open up the bird, you take all the stuff inside out, and you very carefully stuff it with cotton wool that's sterile, and then you sew it back up. You try to make it look as close to sort of a live specimen as possible, or as natural as it would look as a live specimen, and you let it dry. We freeze it to check that there aren't any pests in it, and then we bring it into the dry collections. So again, here we have two of these tufted puffins or crested puffins. You can see their little crests here. And they have these really amazing, in life, these very colorful beaks or bills. And they kind of lose some of that color over time as they age and also as they dry out, as these specimens dry out. But a really cool thing that people have discovered with tufted puffins and kind of with the group of puffins, these pelagic seabirds in general, is that their bills actually fluoresce under ultraviolet light. And we'll try to see if you can catch this on camera. But if you shine a UV light on their bills, it fluoresces kind of greenish blue in certain areas. And it's a little tough to tell, but you get this really cool fluorescence that shines back at you. And it's really interesting, and it's fairly common for birds to have ultraviolet reflectance in their feathers, in their bills, in their body parts sometimes, because they can actually see ultraviolet. So this is something, again, so insects can see UV, and plants have UV, which insects use to guide for pollination, and birds can also see in the ultraviolet spectrum. However, we can't, right? Humans, we can't see UV, mammals can't see in the ultraviolet spectrum. So this was kind of unknown to us until people just started looking at museum specimens and shining black lights on things, basically, to find this really cool fluorescence. No one really knows why this happens, why this phenomenon happens in puffins or in other birds. They think it could be related to sexual signaling, so finding mates in the species, attracting partners, things like that, but no one really knows for sure. Scientists are doing experiments with live birds where they make little sunglasses to put over the eyes so they don't shine the UV light in the eyes of the puffins. And there's some really cool work to be done, but we still don't know. We have a few hypotheses. So that's kind of it for talking about birds right now. We'll move on to some cabinets, and I'll show you a little bit more. We'll keep talking about UV reflectance, but we'll use a different group of animals. So come with me and we'll have a look at some drawers in the compactors. So now we just talked about UV fluorescence in birds and how that's a pretty common phenomenon because they can see that light. They can detect those wavelengths. However, it's virtually unheard of in mammals, but we're going to see an example of a couple of species where they actually do glow even though mammals can't perceive this wavelength of light. So here we have a drawer full of flying squirrels. So these are from the United States, so from the eastern part of the United States. Here in Connecticut, we actually have two species of flying squirrel. Their genus is Glaucomes. You can see some examples of round skins, just like the puffins that we were talking about earlier. Some are kind of spread out as they would be when they're flying. Others were kind of just tucked in as little round skins. But the really, really cool thing with these species, and again, we're wearing gloves as we handle any skins, is that if we take our UV light and we shine it on the belly fur or the ventral pelage, it glows pink. which is really cool. And again, the squirrels themselves can't see that. It's something that birds could pick up or insects could pick up. Let's see a little brighter there. But it's only the fur on their bellies that glows, that reflects this UV light as this like bright pink color. And when you think about it, why would a mammal have something like that? Why would it glow pink in UV if the mammals themselves can't detect it? It's not a signal that they can pick up. And nobody really knows. This is something that people have kind of thought about a while. People have one hypothesis that they're doing this to mimic owls, actually, and it's an adaptation that's kind of just proliferated over time. There are species of owls that exist in the same regions as these species of squirrels, and they actually fluoresce that same color. So barn owls, barred owls are another example, and juveniles of those owls glow that same pink when you shine UV light on them. So some people think that this is just an adaptation that's kind of come about and given certain squirrels an advantage, even though they can't see it. The owls kind of mistake these flying squirrels as they're jumping around. They're glowing, fluorescing pink in the night. And the owls are mistaking them for other owls rather than for prey items for food. And so those squirrels are at an advantage. So again, kind of an interesting phenomenon that only was realized when people started going through museum collections and shining black lights on things. So it's not really something you would think to do, but it's something that people are doing increasingly to see if there are these interesting kind of unseen traits that animals have, and trying to think about why this would even exist. So we're going to move on from flying squirrels and our round skins of those squirrels, and we're going to look at some more examples of mammal specimens here and some other ways to prepare skins here in the BRC. So you can see here, these are jumping mice, Nepeozapus insignis, and you can see we have quite a few of them here. They have very long tails. They also have very long feet, hence the name jumping mouse. They're very, very good at jumping. In fact, they can use their big, strong feet to propel themselves distances of up to 9 feet to escape predators and get around. They're found mostly in kind of moist forests here in Connecticut, but they do range all over the East Coast. So here we have a lot of specimens. You can see they're round skins for the most part. A lot of these little, trying to make them look true to life, kind of spread out their fingers and toes. But we also have a specimen that looks a little bit different, and it's a flat skin. So we also call this a card skin. Basically, the little mammal, this little jumping mouse, has had all of its insides removed, as have all these other round skins, but it hasn't been stuffed with cotton. It's been stretched on a cardstock, an archival material. So there are a few different ways that you can use to prepare mammals and include them in the collections and use them for different scientific studies. You can still take a lot of measurements. You can look at their pelage or their coat color. You could take measurements of their feet, of their tails, of different kind of morphological characters to do a bunch of different studies. And one thing I was mentioning before with round skins, with birds, we keep their skulls intact in the skin. However, with mammals, we usually take it out. And so that's what all of these little files are for. So some of the all of the skulls of these specimens are in these tubes, along with some bones sometimes. Again, we keep some bones in the skins, but each of the skulls is associated with one of the skins. And you can, again, take measurements on the skull that way and associate it with an individual, with another specimen. So those are some examples of how we preserve mostly birds and mammals here in the BRC. We keep those in the dry collections as either skeletal material or round skins. And I'll talk a little bit later about other alternatives that we use for things like our fish or our herpetofauna, and we preserve those in liquid. So we'll talk about that a little bit later. I'm now going to talk about another type of specimen preservation method that we use in the Vertebrate Division, and that's talking about wet or spirit or fluid preserved specimens. So in the BRC, we have a pretty extensive collection of animals, of vertebrates that we keep preserved in very flammable ethanol. So we keep everything at about 70% ethanol, but prior to being put in this liquid, we have to do what we call fix the specimens so that they don't degrade. And this is usually done in another chemical such as formalin, where the tissues, you're kind of fixing a specimen, and its tissues, you don't want them to rot, basically, after it's been put in the ethanol. So once we fix something, we put it into our 70% ethanol, and then we can keep it basically forever in our wet room or in our spirit room. The biggest thing to think about with fluid preserved specimens is pretty much how dangerous it is to keep them. Again, we have thousands and thousands of gallons of ethanol in these different jars that are keeping specimens preserved, and we have a lot of safety precautions that we have to follow. So things like making sure we have really good jars that are very secure and stored securely on shelves when we move them around in our compactors. And we also have a really state-of-the-art fire suppression system in the room where we keep all of our wet specimens. It's called an energen system. And basically, if it detects fire, either through heat, through temperature, or through wavelengths of light that flames produce, which is why we're actually not filming in there, just because it's kind of dangerous to bring cameras into that room, because lights from the cameras can activate the system. So once that system detects a fire, it wants to put it out really quickly to protect everything and to basically prevent a giant fire from raging in that room. So it sucks all of the oxygen out of the room and pushes in a fire suppression agent and really quickly handles the fire. We've never had it been activated, and we hope we never will. But that is a very real danger that presents itself when dealing with fluid preserved specimens. So now I'm going to talk about a few examples of things that we keep in ethanol in our fluid preserved collection. And again, it's most common to preserve fish and herpetofauna, so amphibians and reptiles, in ethanol. We do have some examples of birds, eggs, and mammals that we've preserved in ethanol, but like I told you before, we usually keep those as skeletons or round skins if we have them. The first jar that I'm going to talk about contains a whole bunch of fish. So probably 100, maybe 100 plus specimens of what we call pygmy sculpin fish, so Caudus paulus. And this fish is really small, but really interesting because it's only found in one spring in Alabama. So it's found in a spring called Coldwater Spring, a little bit to the east of Birmingham in Alabama. And because of this really restricted geographic range of this species of fish being found only in this one pool, it's listed as very critically endangered and is susceptible to pretty quick extinction through stochastic events like human-induced pollution. These were collected, I think, in the 70s. You probably couldn't get away with collecting this number of individuals from that spring these days. But it's really valuable that we have these in our collection and people can conduct studies on a lot of different individuals of this really rare species. So, as I mentioned before, we also have a lot of herpetofauna, or herps, that we keep preserved in ethanol. This is a hog-nosed pit viper, so a venomous snake from Central America. It's kind of small. They're described as sit-and-wait predators, and that means they basically sit on the forest floor in the rainforest, coiled up, waiting for prey to pass by. And they detect their prey, they bite it, and it gets envenomated and killed. So they're kind of coiled up on the forest floor, much like this little guy is in the jar. And again, this is a really common preservation method for snakes, amphibians, lizards, things like that as well. So we have that. And then here are a couple of examples of mammals that we've preserved in ethanol. These are two examples of armadillos I think these were collected in Paraguay. They've both been preserved in ethanol, and you see the level of the fluid. We always try to keep it so that the entire specimen and the tag, the data associated with it, are submerged in that ethanol. So this is the Pichi ciego, or the pink fairy armadillo, and this is the greater fairy armadillo. Both of the species are found in kind of arid regions in Argentina and Paraguay, and they're very elusive. They're very difficult to see. They're very secretive. They only come out at night, and almost nothing is known about either of these species. Even less is known about the greater fairy armadillo compared to the Pichi ciego. So we've preserved these guys in ethanol, and if you look closely, you can actually tell that their skulls have been removed. So like I mentioned when we talked about the round skins of the mice, we remove their skulls from the round skins. These liquid preserved specimens also had their skulls removed, and they're kept separate so people could do measurements on them as well. So now we're going to talk about kind of an unpleasant subject, but one that's very important to the study of biodiversity and ecology. And we're going to be talking about and showing some examples of extinct organisms. And here we have 4 different species of birds, and I'll tell you some stories about each one. So the first example that we have here is this really lovely taxidermied specimen of the passenger pigeon, which you can see here is extinct, eliminated by man. And this is as with all of these, a fairly sad story. This species was once thought to be incredibly, incredibly abundant, probably one of the most abundant species of birds native to North America. It was found all over the United States, mostly in the east too, and would migrate in these really, really big flocks to feed and to mate, to find breeding grounds in forested areas. The passenger pigeon was so numerous that people basically thought they could kill it for fun, they could eat it for meat, shoot it for sport, but it would never ever be gone because it was so abundant. Some people do think there were between 3 and 5 billion individual birds at one point in time, and you can kind of compare that with how many humans exist on the planet these days. We're at about, I think, 7.8 billion these days. That's a lot of passenger pigeons. So the fact that this bird went extinct was kind of a wake-up call for people, and it was driven extinct by humans. So again, hunting and also deforestation, so loss of breeding grounds, really, really contributed to the rapid decline and ultimate extinction of the passenger pigeon, declared extinct officially in 1914 when the last captive pigeon, Martha, at the Cincinnati Zoo died. So the last wild bird of the passenger pigeon was shot in 1901, and Martha died in captivity in 1914. So a really sad story. Again, people just kind of went out, killed these guys with reckless abandon, and eventually drove this species to extinction. Moving on to another, probably not quite as famous as the passenger pigeon, but I think a really nice charismatic species that was native to the United States is the Carolina parakeet. So here, one of our native US parakeets, again, very, very abundant in kind of prior to the 1800s. It was hunted prolifically, more for sport than anything, and its numbers really, really declined in the 19th century. And what's really interesting about these birds, again, this is a native bird to the US. It's very brightly colored parakeet, and it's a little bit more dull in this taxidermied specimen, but still very bright. And people actually thought that the coloration of this bird meant that it was poisonous. And this is because this species of parakeet would really eat this Xanthium strumarium plant, these little spiky fruits. It would open up those fruits and eat the seeds inside. And the seeds, the common name for this plant is cocklebur, and the seeds are very, very toxic to mammals especially. They can contain a really high doses of a glucoside poison, and the birds weren't affected by this poison whatsoever. So they would just go crazy eating these fruits and potentially sequester poison in their feathers. So people thought they were poisonous based on not very many anecdotal accounts of birds eating Carolina parakeets and then dying. So Audubon was one person who had an account of his cat dying after eating a Carolina parakeet. No one's really done any studies to see if the birds could sequester these poisons in their feathers and then have this kind of warning coloration to show off how poisonous they were. And it's a little difficult now. Our specimens are fairly old. I don't know if you could actually do any kind of chemical analyses on their feathers, but some species in Papua New Guinea, some birds from the Pito Hui genus are actually poisonous. So poisonous birds do exist. They're out there, and maybe the Carolina parakeet was one of them. Unfortunately, we don't know. It was driven to extinction with the last captive parakeet also dying in the Cincinnati Zoo. His name was Incus, and he was kept in the same cage as Martha, the last passenger pigeon. And Incus died in 1918, 4 years after Martha did. So again, that species was lost with the death of Incus. So moving on here, we have this really lovely specimen of a heath hen. And heath hens were native to the eastern coast of the US. All of the extinct birds we're talking about today were native in the United States. And basically people treated them like little turkeys. and really heavily hunted them for their meat. So they're pretty round, they're fairly potato-shaped, and people really liked to eat heath hens for their meat. So again, this is another example of a species that was very, very abundant, but heavily, heavily hunted. So by around the mid-1800s, the population came to only about 100 or 200 birds left in the wild. And the heath hen is a really interesting example of a species, and among the first species, that humans actually tried to prevent from going extinct very actively. So people passed anti-hunting legislation and tried to establish land preserves to protect the little bit of habitat that was still there for heath hens. But unfortunately, it was just too late, and those attempts were ultimately unsuccessful, but they did help other species of birds, so not all was lost. However, the heath hen was officially declared extinct in 1932, probably well after the last wild bird had been killed. And then moving on finally to this last specimen here, which is known as the Eskimo curlew. It has a lot of different common names, actually. My favorite is called the dough bird, D-O-U-G-H bird, named because it was really, really fat whenever it started its migration. So again, this bird, this curlew, was thought to be among the most abundant shorebirds in North America. Are you kind of getting a sense of a pattern here? This bird was very, very abundant, but was very prolifically hunted, again, just like with all those other three species, with some people taking, or kind of every year, up to 2 million birds were killed per year in the late 1800s. So again, a once very abundant species was heavily, heavily hunted, basically to extinction. Now, the interesting thing with doe birds is that they haven't yet really officially been declared extinct. So there was a reliable sighting of an individual of this species in 1963 in Alaska. And although it hasn't been declared officially extinct, it's listed as critically threatened and possibly extinct. So it hasn't been cited for many, many years, and especially cited reliably by people who kind of know what they're looking for in this species. So yet another example of a species that was driven to extinction by humans in the late 1800s. All right, so that's going to do it for today's tour of the vertebrate collections. Thanks for joining me today, and I hope to see you in real life in the future after the pandemic. Thanks a bunch.